I want to pick and choose some things here that I want you to know. Okay. There are a couple slides that I have in here. I thought I wanted to go into it. But I'm not, in retrospect, I don't have time and I don't want to get into it too much. So there's going to be things I pick and choose here. So hopefully you're paying attention, you're listening, you're highlighting, watching my magenta colored pen here on your screen here. Where does all this energy come from? Where does all these things come from? Again, our diet. We just mentioned that in 19. Glucose, glycogen, lactic acid. That's what we've kind of talked about so far. All of this is associated with this molecule of carbohydrate, or this biomolecule of carbohydrate. Okay. There are some terms that you should know. Because when we start talking about muscles and whatnot, we'll hit these words again, and you'll need to kind of understand their ability to utilize oxygen, so their ability to go back and get into cellular respiration and provide all of these units of energy for which that these skeletal muscles can contract, okay? Because that's where we talk about it the most. We have a little bit of stores, okay, of glucose. Not a whole hell of a lot, okay? Predominantly, it's stored longer term as glycogen. Okay. The reason why is because of 1A there. Okay. If we're going to move glucose, we're going to be moving a lot of water as well. Okay. And we may talk a little bit about that in digestive system, maybe a little bit. It's something that we talk about a heck of a lot more in nutrition, to be honest. Okay. But to get glucose to enter into a cell, I need sodium, and for sodium to move across, I need water. So I need a lot of water if I'm going to have a lot of glucose coming across the cell membrane into the cell, right? So for that reason alone, I don't have a huge store of glucose inside these cells because I'd have to pull across a hell of a lot of water. So we don't pull across basically what we need. Okay. There's a creatine phosphate system, and that's like maybe 2 or 3 seconds worth. So, there you go. So that's not a whole heck of a lot of glucose. We store glucose as glycogen, and once we convert it into this long-term storage of glycogen, then I can remove this concept of bringing across a lot of water. I can convert it, and then I can get rid of that water, right? Where do I store glycogen? 3 places. 2 of them, predominantly more than anything, the liver, skeletal muscles. Okay. Yes, cardiac muscles too. Okay. That would be number 3 on this list. But generally, when you think of activity, you think of liver and you think of skeletal muscle. All right. Even though cardiac muscles start contracting, week 5-ish, and don't stop until you're dead, that's probably a hell of a lot of activity. Okay. So that's why they're listed here. All right. But we're going to be talking about it in terms of skeletal muscle function, not cardiac. Cardiac muscle has a lot of other working parts that we'll look at. Glycogen is formed from glucose via glycogenesis. What does glyco mean?
Carb.
Yeah, glucose. Yeah, carbohydrate. What does genesis mean?
To make.
Formation of. Okay. Is glucose simple or complex?
Simple.
Beautiful. Simple, you betcha. It's a monosaccharide, right? Glycogen is hundreds or thousands of glucose molecules strung together. So glycogenesis is the formation of glycogen, carbohydrates, okay, stringing all of those glucose molecules together in this very crazy, highly branched storage form. Because it's highly branched, I can easily hydrolyze it and break it down and get those glucose molecules to enter back into the blood and magic, more glucose. Okay. What is glycogenesis? Formation of glycogen. Glyco is sugar. Okay. The basic monomer of carbohydrates is glucose. So this is a process that happens in the liver, skeletal muscles, cardiac muscle, where I string hundreds or thousands of glucose molecules together into this storage form. How does this body know to convert glucose into glycogen? Can anybody give me as a reason as to what you would think? How does it know?
Can't glycogen be used a lot? Well, it can be stored easier, and so I feel like the body would be like, hey, we have something that we can't store a whole lot of. Let's convert it into something that we know how to store and then use like better.
Okay. But how does it know?
I don't know.
Receptors, right? Receptors are how these cells communicate. They don't have a language, right? They don't have a mouth. They don't say, hey, you know, I'm full of sugar. I'm content. You need to take this glucose and take it someplace else. It doesn't do that, right? So how do they communicate? They communicate through receptors. So we're going to have receptors that are going to be present in cells, on cells, I guess I should say. They're going to help bring all this glucose across. And again, we have receptors that monitor sugar levels in the blood. When my sugar levels are low, these cells are going to show all of these glucose receptors on the surface of it, being like, hey, I'm hungry. I need ATP. I need to do stuff. I need sugar. Body's going to sense that. Okay? We have chemoreceptors detecting for that carbohydrate level. Body picks up that, communicates to the pancreas, okay, and gets hormones involved that gets that glycogen out of storage. Sugar level starts to rise, gets to 100, gets a little over, and now these cells will start to downregulate. It'll take away those receptors. When there's no receptors communicating I need sugar, now the body picks that up. There's no receptors there. Body doesn't need this sugar. So now it switches gears. That pancreas secretes another hormone that says, hey, go into the closet. We're going to store you for long term. And then we'll take that up into some other cells, liver cells, predominantly, followed by skeletal muscles, cardiac muscle. And we'll now convert those glycogen molecules into, those glucose molecules into glycogen that are there. And now I have some long-term storage. But it's all done with receptors. All right? Chapter 6 starts to look at that phenomena of communication with receptors and how they bring stuff in and get rid of stuff from a cell. Okay? So this is just our first dip of the toe into chapter 6, okay, in that next unit. Glycogenolysis. What's lysis mean?
To break down.
Mm-hmm. Yeah, to break down. Break, DWN. Okay. Breaking down what? Glycogen. So glycogenolysis is what happens when I want to get glycogen out of storage so that the cells can utilize the glucose molecules. Okay. So when the cell breaks down, glucose, it breaks down that glycogen, getting glucose to flood the blood vessels, enter into the cells, and start doing work. Okay. I'm not going to ask you the first step, fructose 1-phosphate. Okay. It enters into the pathway. Glycogen does. The first pathway is glucose 1-phosphate, glucose 6-phosphate, and then from there, boom, into the blood is glucose. Okay. I don't want to get into all that. I don't want to get into the explanation of that because that again is biochem, some nutrition. Okay. If you're more curious, we'll talk about it a little bit more there. I'll save you the trouble. Cross off slide 36. Okay, cross off slide 36. I won't ask anything off of this slide. I do want you to know a couple of things here, and we'll go through the next couple of slides. We'll take another little break. Okay. Cori cycle happens in the liver. Okay. Cori cycle occurs only in the liver. Cori cycle is what happens when I produce too much lactic acid. Or you could say I have a lack of sufficient amount of oxygen. Okay. When I produce too much lactic acid, the cell converts that pyruvate and that NADH molecule, like I said, into lactic acid. Lactic acid is then removed from the cell, dumped into the blood, and the red blood cell takes that lactic acid and ultimately it drops it off in the liver. Red blood cells can utilize it. So it's not like it's not doing anything. So it'll provide some stuff for the red blood cell. But the liver is the only dude that has the enzymes involved to help convert lactic acid into glucose. So here within the liver, the Cori cycle takes over. Right? Cori cycle takes lactic acid. The liver takes that lactic acid that you've built up, and we can convert it back into glucose. Glucose enters into the blood and ta-da, enters into glycolysis there, and I get 2 whopping ATP out of it, right?
Converts it back into glucose?
It does.
So lactic acid to pyruvic acid and then back to.
Yep. There are a number of steps. Okay. So here's lactic acid. I got to convert it back to pyruvate, takes it back to glucose 6-phosphate, which then we convert within the bloodstream to glucose. Okay. So lactic acid gets delivered to the liver. And that liver starts the process of converting that lactic acid ultimately into the GLU. Okay. All the individual steps we're not getting into. Okay. But that liver can convert that lactic acid ultimately back into glucose. Glucose can enter back into the blood, be delivered back to skeletal muscles, and again, can provide units of energy. Okay. Where does the Cori cycle happen? Liver. What does it do? Converts lactic acid back into glucose. When is this system functioning? Whenever I have lactic acid produced. Okay. Generally, this is the most common pathway when it's in an anaerobic state. Okay. When I don't have enough oxygen. Is lactic acid always evil and bad?
No.
No.
Well, no.
Sure sounds like it. Why do you think it's not?
Well, I mean, this is an example, right?
This is an example. Okay. But even greater than that, lactic acid, by definition, right, changes the pH of something, right? It's an acid. It's becoming more acidic, right? So wouldn't that change the pH of the tissue that it's within? Sure. I mean, acids accumulated in hydrogen ions causes the pH to start to drop down. And, we were told at the beginning, unless I've been fed lies, that, every tissue has its optimal pH. You kind of value that it functions properly within. Yes. In the, I think, 60s, I think somewhere in the 50s, 60s, early 70s, I can't remember. pH from that lactic acid, okay, oh my gosh. Yes, lactic acid causes a drastic pH change. And what happens if you run for a really, really long time and try to maintain that pace? All of a sudden, the muscles start cramping up. So through that little basic study, lactic acid is detrimental and it causes skeletal muscles to stop functioning. You cramp up, you stop. But they've discovered that lactic acid, this has been something that we've been dealing with ever since we started with skeletal muscles on our bodies and moving around. You are producing lactic acid a lot of times, even in a slightly, more aerobic state as well. Okay, so you're always getting some production of this lactic acid. What they've discovered is with athletes, if you get into that zone where you have intensity and you don't necessarily have as much oxygen coming in, okay, you're pushing yourself, right? That if you kind of get to that, almost to that wall and hit it, you get this lactic acid buildup. The more you get into those zones, those high intensity zones of exercise, the more adapt your skeletal muscles get at handling lactic acid, using lactic acid to make energy, to maintain that high level of activity, that intensity level. And those that are able to get into that anaerobic zone and stay there for longer periods of times, their skeletal muscles become more efficient at it, at processing that lactic acid. What does that mean? That means they become more adept at utilizing the oxygen more effectively. So you become more aerobically fit in the process. So this whole concept of lactic acid being the devil in terms of skeletal muscle function is really not valid anymore. And that's like just 3 or 4 years old. What were you saying, Sam?
It's just kind of a long-winded way of saying like they become cramp resistant.
Yeah.
From the lactic acid.
Right. And that's only because they become more efficient at utilizing oxygen. So it takes them longer to get into that anaerobic state.
So does the Cori cycle only happen when it's in excess of lactic acid or when it's present?
When it's present, lactic acid will be shipped there.
Okay, so it's not in excess, it's just...
Yeah, if you're much more effective at utilizing your oxygen and being more efficient with your skeletal muscle contractions, you don't produce as much lactic acid. So the Cori cycle is not going to be this huge production of 2-whopping ATP. You're getting enough oxygen in. Okay.
Okay.
But the only place that I can handle lactic acid is in the liver. It's the only dude that has the enzymes in which to break it down and convert it back into glucose.
Right. Okay. So when lactic acid is present, no matter what, the Cori cycle will happen. It just might not be as quote unquote intense, depending on like how adept your skeletal muscles are at handling it.
Correct.
Okay.
Yep. Now let's say you've gotten up and run, Sam. Let's say you can run 3 minutes without getting winded. Okay. We're just playing around. All right. So you get to that 3-minute mark and oh my gosh, you want to keep going and pushing past it. but you start cramping up a lot, okay? And you get that stitch in your side, right? Well, that as you breathe, those muscles are contracting, right? Well, now those muscles are, now you're breathing more. So those muscles are contracting more around that rib cage, diaphragm, external and internal intercostals, all those things, okay? So you're going to be producing a whole hell of a lot of lactic acid. There's a lot of muscles involved in that process, right? So you're going to start force breathing yourself. Those muscles are going to be engaged more. You're producing a lot more lactic acid, okay? So you're going to be producing a lot. It's going to be too much for the body to do, okay? You're not going to be getting enough glucose converted to get back to those muscles so you can continue to heave ho like that. Whereas somebody who can go 30 minutes at a time, they don't produce as much lactic acid. They still send it to the liver, but they're getting enough oxygen in.
That it doesn't have to take that lactic acid. And
yes.
Okay.
So this Cori cycle is not going to be a huge thing. Okay. So these are the two terms that we used, and these terms make good multiple choice questions. So that's where you would see them. Okay. Glycolysis, we talked about that. There's glycogenolysis, glycogenesis, formation of glycogen, breakdown of glycogen. We haven't talked about this one yet, even though glucose is in the word. This is where we use something other than glucose to make glucose. Okay. And I guess, lactic acid could be that. But this is usually when we talk about, amino acids. You have some amino acids that could be converted back into glucose. So that's called gluconeogenesis, a new form making glucose, GLU, okay? A new form of glucose. It's basically glucose from a non-glucose source. Lactic acid could be that. Amino acids, proteins can be involved in gluconeogenesis. Okay? And again, I have the machinery for that in the liver. That's where we see gluconeogenesis occurring. The other three we haven't talked about. They're next. Okay, but let's take a little break. Let me pause.